Dr. Libby, are you hearing me? Yeah. My name is Flavio Fuchs. I'm coordinating this section that's been the last section of this Symposium, International Symposium of Cardiovascular Epidemiology, led by Dr. Sandra Fuchs. They have talked with you many times about this symposium. I would like to thank you very much to having accepted our invitation to participate. We're going to give the last speech in the symposium that is being conducted in honor of Dr. Jorge Pinto Ribeiro. You are here with Cora and other family members that are attending to this section. We had very nice presentations before, and I'm absolutely sure that we're going to have a closing conference of high quality. In order to introduce you to our audience, I would like just to say, who is Peter Libby, for those who don't know? Well, he's just the chief of cardiology of the best medical institution in the world. Nothing more needed to be said about. Please, Dr. Libby, you have the word.
Well, I would say it's a terrific honor to represent the Brigham and Women's Hospital at this symposium dedicated to celebrate the life of a great man. Jorge Pinto Ribeiro was a fantastic doctor. He was a wonderful researcher. He was an educator. He was an athlete, and he was, above all, a generous and kind mentor and man. So you said in your very kind opening remarks that the Brigham and Women's Hospital is a top medical institution. I would say that it is our tradition of welcoming foreign scholars to the Brigham and Women's Hospital, and having a two-way learning experience with them is one of the strengths that makes us a good institution. And we were very privileged to have Jorge Pinto come and spend time with us as a trainee back in the 1980s. And it has been a particular pleasure for those of us in leadership here at Brigham and Women's Hospital to see how he was able to take the learning that he did here and the values that we share and create such a wonderful cadre of people in Porto Alegre. And it was wonderful to learn just now that the Clinical Research Center will be dedicated to him so that his memory will remain with us in perpetuity. So one of the aspects of the Brigham and Women's Hospital that we're very proud of is our ability to conduct translational research, where we have a tight connection between the basic science laboratories and our clinical investigation. And so what I would like to share with you this afternoon is a way in which we have attempted to take some very basic science and bring it to patients. And in the title that I gave to the organizers, I think I said going global, because when we conduct the clinical research, we are fantastically privileged to work with investigators all over the world, including Brazil. So the story that I'm going to tell you is about inflammation in atherosclerosis. Most of my talk will be forward-looking with what's recent and where we're going with this field. For those who are interested in a historical overview, you can consult this article, which tells the history of about a quarter century of work on inflammation in atherosclerosis. But as an introductory or prefatory remark, I would like to call attention to one of the figures from this recent article. And it makes a very important point, and that is that inflammation does not supplant or demote traditional risk factors. What it does is to provide a mechanism that links them to altered arterial wall function. So the inflammation biology that we've been concerned with in the laboratory doesn't say that LDL, angiotensin II, obesity, smoking, or other risk factors are not important. It just gives us some mechanistic insight into how these risk factors can evoke inflammation and thus alter the behavior of the artery wall in a way that gives rise to the disease. So the inflammatory hypothesis is not in competition with the lipid hypothesis or other traditional risk factors. Now, a little more than 10 years ago, we summarized some of the steps where inflammation participates in the initiation of atherosclerosis in this cartoon that was published in Nature. And we posited roles for inflammatory mediators in the adhesion of blood leukocytes. In the atherosclerotic process, most numerous cells are the monocytes from blood. The chemoattractant molecules call them into the artery wall. There are factors that cause the monocyte to mature into a macrophage, to express scavenger receptors that allow uptake of modified lipoprotein particles and the creation of the macrophage foam cell, which is the hallmark of the early atherosclerotic lesion. And once these foam cells are recruited to the arterial intima, they can give rise to many mediators that will then cause the progression and complication of the disease. This particular slide mentions just the mediators that my laboratory has worked on. Vascular cell adhesion molecule 1 is a typical adhesion molecule. Monocyte chemoattractant protein 1 as a typical chemoattractant. Macrophage colony stimulating factor as a stimulator of scavenger receptor expression and maturation of the monocyte into a macrophage. But this slide is oversimplified because there are many other mediators that have been worked on all over the world, including in Brazil, and also because of the implicit assumption that all monocyte macrophages are created equal. Since we published this in 2002, one of the hot areas of atherosclerosis research has been the recognition that monocyte and macrophages are heterogeneous in atherosclerosis. This issue of the Journal of Clinical Investigation was entitled "Macrophages Cause Metabolic Mayhem." And it contained a pair of articles from my laboratory and from Gwendolyn Randolph's laboratory calling attention to heterogeneity of monocytes in hypercholesterolemia. Our pair of papers was accompanied by an editorial by a pioneer in this field, Professor Simon Gordon, and he was able to summarize the markers that we used to discern two classes of monocytes. Monocytes that bear high levels of the surface marker LY6C have particularly pro-inflammatory functions. This is the index observation from our laboratory, from my then postdoctoral fellow, Phil Swirsky. When we make a mouse that is deficient in apolipoprotein E and thus will develop hypercholesterolemia on a Western type diet, an Occidental type diet, there is a exponential increase in the number of these pro-inflammatory monocytes in the peripheral blood and in other tissues, including the spleen. We soon found that the source of these blood monocytes that are particularly pro-inflammatory, that increase in the hypercholesterolemic mouse, they derive from the spleen in a preformed pool in the subcapsular splenic red pulp. if we stain the spleen of a normal cholesterolemic mouse or a hypercholesterolemic mouse with an antibody that recognizes a marker of myeloid cells, you see the accumulation of these inflammatory cells in the spleen of the hypercholesterolemic animal compared to the normal cholesterolemic animal. We determined that the spleen is a site of expansion of these pro-inflammatory monocytes through extramedullary hematopoiesis. And now the postdoctoral fellow of my former postdoc, with who I still collaborate in these interesting experiments, found that extramedullary hematopoiesis generates these pro-inflammatory monocytes, and they can take up residence, they can infiltrate the atherosclerotic lesion. So I'm not going to show you all of the data which are published in this recent paper, but the concept is that we have expansion of progenitor cells in the spleen to enlarge a reservoir of pro-inflammatory monocytes, and some of these monocytes find their way through the bloodstream to the atherosclerotic plaque, where they can mature into macrophages that drive the progression and complication of this disease. So this has been a nice adventure for us in study of mouse atherosclerosis, but do these new insights into plaque inflammation have clinical implications? I would like to describe to you a hypothesis that we developed several decades ago that has recently been validated in the experimental laboratory. The concept that we put forth a number of years ago was that an echo in the arterial wall of inflammation from a remote source, systemic or local, for example, acute myocardial infarction, could actually aggravate atherosclerosis by stimulating a wave of inflammation in the plaque. This is a very old paper from 1991 where we described the expression of a pro-inflammatory mediator known as interleukin-1 in the artery wall when we gave a rabbit, in this case, a model inflammatory stimulus systemically. We injected gram-negative endotoxin intravenously, and we saw a burst of interleukin-1 gene expression in the rabbit aorta. Back then, we were thinking of endotoxin as a model stimulus. That was before we had today's appreciation of the importance of the microflora, the microbiota in the intestine, and impaired intestinal barrier function that might actually make gram-negative endotoxin more than a laboratory artifact, but a pathophysiologically relevant systemic inflammatory stimulus. Our next step was to create graded levels of atherosclerosis in a series of rabbits by feeding them increasing concentrations of cholesterol in the diet. So we kept rabbits on a low cholesterol diet or supplemented it with 0.1% cholesterol, 0.3% cholesterol, or 0.9% cholesterol, making a graded amount of atherosclerosis in the aorta. We then used the optimum concentration of endotoxin and analyzed at the optimum time as shown in the previous experiments, and tested the hypothesis that there would be greater inflammatory gene expression in the animals that had the greater burden of atherosclerosis, in whom the inflammatory cells were lying in wait in a quiescent state, a relatively quiescent state, in the preformed atherosclerotic lesion. And when we gave the systemic inflammatory stimulus, intravenous endotoxin, we tested whether there would be a greater burst of pro-inflammatory gene expression. And indeed, as you can see, as we increased the amount of cholesterol in the diet, we had higher levels of interleukin-1 beta gene expression in the aortas. So the systemic inflammatory stimulus evoked a local inflammatory response that was proportional to the amount of pre-existing atherosclerosis. Now let's fast forward to last year, when using modern techniques, we were able to show that myocardial infarction accelerates atherosclerosis by ligating coronary arteries in hypercholesterolemic mice that had, just like our rabbits back in the 1990s, preformed atherosclerotic lesions. Now, what disease-relevant inflammatory function did we monitor to test whether myocardial infarction causes an echo of inflammatory activation in the pre-existing atheromata? Today, in the New England Journal, a paper is published which covers 2 decades of work from my group that I'm not going to talk about today, but that explains why we chose protease activity as a disease-relevant readout of inflammatory activation. Because we have a body of work reported in today's New England Journal of Medicine that implicates proteinases that are expressed by the foam cells in the plaque as the culprits in degrading collagen that lends strength to the plaque's fibrous cap and protects the plaque from rupture. So protease activity is an index of activated macrophages that is related to the acute coronary syndromes. So using molecular imaging technologies that we have helped to develop over the last several years, we are able to ligate a coronary artery in a mouse which has low levels of protease at baseline, shown by the cool colors in this scale of pseudo-colored proteinase activity. But 3 weeks after a myocardial infarction has been performed, there is greater protease activity in the aortic root. We can quantitate that in a series of animals here, and we can excise the aorta and show greater proteinase activity after 3 weeks in the aortic root of the animals that have had coronary ligation than in the controls that have pre-existing atherosclerotic plaques but have not had coronary ligation. So here's the increase in proteinase activity. So acute myocardial infarction amplifies local inflammatory activation in the pre-existing plaque. Myocardial infarction also causes an increase in the accumulation of the pro-inflammatory monocyte subset, as shown by the accumulation of the LY6C high monocytes after myocardial infarction compared to the natural history of accumulation of monocytes in the atherosclerotic lesion in these hypercholesterolemic mice. There are important structural consequences of this overexpression of proteases after myocardial infarction. The plaque's fibrous cap becomes thinner, as you can see here, a very thin fibrous cap, compared to the thicker fibrous cap in the animal that did not have the myocardial infarction. So we have a thick fibrous cap here and a thin fibrous cap after myocardial infarction. That is quantitated here, a thinning of the fibrous cap after myocardial infarction. And there's a corresponding increase in the necrotic core area following myocardial infarction. These are changes that would be expected in human terms to render a plaque more vulnerable to rupture and cause a thrombotic complication. So the new insight that these experiments have provided for us help us to understand why, or one of the reasons why, there is an early recurrence rate of acute coronary syndromes after myocardial infarction, because myocardial infarction can enhance atherosclerosis. In this paper, but I haven't had time to go into it with you this afternoon, we actually found that sympathetic nervous system activation by stimulating beta-3 adrenergic receptors can mobilize progenitor cells from a bone marrow niche, providing the substrate for extramedullary hematopoiesis in the spleen and hence expansion of the pro-inflammatory monocytes in the plaque. So this is our historical understanding of atherosclerosis, that atheroma gives rise to myocardial infarction. What I'm telling you this afternoon is that myocardial infarction can actually activate the atherosclerotic plaques. And there are very interesting detours through the brain, the bone marrow, and the spleen that really provide us with a wonderful example of truly integrated physiology. So we believe that atherosclerosis involves inflammation at all stages, from initiation, the progression, and ultimately the thrombotic complications. And we've traditionally considered atherosclerosis as a logical temporal progression from initiation to complication. And the news for today is that actually the complication can feed back on the processes that cause the progression of the plaque and can push the evolution of the atheroma. It's been my great privilege in these recent studies to work with my former postdoctoral fellow and current collaborator, Phil Swirsky, and his scientific partner in this work, Matthias Narendorf, a very talented cardiologist. Well, I promised translation to the clinic, and I've given you conceptual ideas about how inflammation biology can translate to the clinic, but how do we take the experiments performed in rabbits and in mice and transfer them to humans, who are, of course, our patients? And the question that arises from all of this new biology of inflammation and atherosclerosis is, can targeted anti-inflammatory therapy reduce cardiovascular event rates and prolong life? That question is the subject of two large-scale clinical trials which are being initiated here at the Brigham, being led by my clinical collaborator, Professor Paul Ridker. And Professor Ridker came up with this schema, a general schema of the cardiovascular inflammation reduction trial. It would enroll individuals who survived a myocardial infarction who are on a full complement of care medications, including high-dose statin, but who have persistent evidence of inflammation defined in some way, randomized to placebo or to a specific anti-inflammatory intervention, and then follow up for hard endpoints. So, which agents would one want to study? Well, a work from Dr. Ridker and my other colleagues and collaborators here at the Brigham and Women's Hospital established for the first time that statins have an anti-inflammatory effect. The statins can decrease biomarkers of inflammation, such as C-reactive protein or interleukin-6, but they do so in part indirectly through a lowering of LDL, and in part, we believe, through a direct anti-inflammatory effect that is independent of changes in the lipid profile. And while we have published a number of statistical deconvolutions to try and sort out how much of the anti-inflammatory effect is direct and how much indirect. These statistical studies will never be able to rigorously separate these two pathways to limiting inflammation. Now, there are some direct anti-inflammatory therapies that have revolutionized other specialties. For example, rheumatology, dermatology, inflammatory bowel disease. The TNF inhibitors have revolutionized the treatment of rheumatoid arthritis. Why are they not the candidates for studying as direct anti-inflammatory therapies in atherosclerosis? For two reasons in the case of the TNF inhibitors. When they were applied to patients with heart failure, they not only caused no benefit, but they actually caused a trend towards hazard. And they also cause an adverse effect on low-density lipoprotein. They raise this traditional risk factor, and therefore we would not want to use tocilizumab or infliximab, the anti-TNF or anti-IL-6 strategies, in our patients at risk for heart failure or cardiovascular disease. So the trial that has begun in the last month or so, that is led by Dr. Ridker, is using low-dose weekly methotrexate, a treatment which I will say our colleagues in São Paulo were able to show could limit atherosclerosis in the rabbit. Low-dose weekly methotrexate has no effect on the lipid profile, but is profoundly anti-inflammatory. The observational data on patients with rheumatoid arthritis who were treated with low-dose weekly methotrexate shows that there is a consistent decrease in the hazard ratio of cardiovascular events in this population, which, as you know, is at heightened risk for cardiovascular complications. However, these observational data are, of course, confounded. This was not an a priori pre-specified hypothesis of these studies, and the cardiovascular events were not adjudicated by the standards that we demand in our cardiovascular endpoint trials. So, published online in the last few days is the design of the cardiovascular inflammation reduction trial, a test of the inflammatory hypothesis of atherothrombosis. This paper was led by my colleague and faculty member Brendan Everett, and this work has been directed by Dr. Ridker, and I'm privileged to serve on the steering and executive committees of this study. The second trial is one that is much closer to the line of research that I've conducted in my personal laboratory over the last decades. And that is targeting a primordial pro-inflammatory cytokine that we have already discussed, interleukin-1. Interleukin-1 has its name because the discoverers thought that it was a mediator that signaled between leukocytes, hence interleukin. In some of our very early work, we were able to show that vascular wall cells, in this case human endothelial cells, could, when exposed to a pro-inflammatory stimulus, again modeled by endotoxin, could accumulate the message that encodes this potent pro-inflammatory cytokine. So not only do leukocytes make this pro-inflammatory mediator, but cells that are present in the atherosclerotic plaque, when exposed to an inflammatory mediator, can express interleukin-1 beta. We rapidly found that IL-1 can induce its own gene expression, providing an amplification loop. These are just references to these quite old studies showing in smooth muscle cells and in vivo in rabbits and in human mononuclear cells in vitro that IL-1 can induce IL-1. IL-1 also can amplify its downstream actions by inducing another pro-inflammatory cytokine, interleukin-6, which is a key mediator of the acute phase response. So IL-1 can induce IL-6. Here's just one data set, just focus here on panel B, which shows on a logarithmic scale the huge amounts of interleukin-6 that are secreted by, in this case, vascular smooth muscle cells that are exposed to interleukin-1. So there are two amplification loops operating in the plaque. IL-1 induces IL-1, and IL-1 induces IL-6. Why is that important? Because IL-6 is the trigger for the acute phase reaction. Many acute phase reactants are pivotally involved in atherothrombosis, such as fibrinogen, the precursor of clots, and plasminogen activator inhibitor, the major endogenous blocker of our own intrinsic fibrinolytic enzymes. So IL-6 tells the liver to decrease production of housekeeping proteins like albumin and increase the expression of these acute phase reactants, which are in the causal pathway for atherothrombosis. The acute phase reactant C-reactive protein is probably not directly involved in thrombotic complications, but it is, for a variety of reasons, a much better biomarker of inflammation than these acute phase reactants. And we can measure C-reactive protein conveniently in the peripheral blood as an index of inflammatory status. Interleukin-1, like many proteins that are pivotal in biological control, has multiple levels of control. IL-1 is first synthesized as an inactive precursor, and it requires limited proteolysis by interleukin-1, beta converting enzyme, also known as caspase-1, to become activated. This is a concept familiar to all cardiologists, and actually I would say that this biochemistry owes a great deal to Brazilian researchers, such as Rocha Silva and Sergio Ferreira, who were among the first to understand that angiotensin 1 requires proteolytic activation to become the active hormone angiotensin 2. The situation with interleukin-1 is just the same. The pro-interleukin-1 beta requires a specific proteolytic cleavage by caspase-1 or interleukin-1 beta converting enzyme to become the active cytokine. Why is this important in atherosclerosis? Because the inflammatory cells in the atherosclerotic plaque are overexpressing this converting enzyme. Here, in studies that we published in the mid-1990s, is the demonstration by this pink immunostain of the expression of this interleukin-1 beta converting enzyme in a foam cell in the atherosclerotic plaque. And this paper reports also the message and the protein. What we didn't know in 1995 is that caspase-1 is the active part of a large supramolecular complex known as the inflammasome, which senses danger signals such as pathogen-derived molecules or crystals, as in gout crystals, and activates interleukin-1 beta activation. So the inflammasome activates caspase-1 and hence formation of mature IL-1 beta. Activators of the inflammasome include every type of microorganism, and there are also sterile activators. I mentioned gout crystals, but recent work from 2 laboratories showed that cholesterol crystals can also activate the inflammasome. Work from Eike Latz, work from Petr Kovenen's group in Helsinki showed that cholesterol crystals activate the inflammasome. Cholesterol crystals are abundant in the atherosclerotic plaque, as you all remember. They're very pretty, they're in needles, they're in plates. The macrophages engage in a death embrace with these cholesterol crystals, as shown in this study from my former postdoctoral fellow, Yongzhang Geng, who did the caspase-1 localization study with me. If you look by electron microscopy in the cytoplasm of monocyte-derived macrophages, you will see cholesterol crystals. So the cholesterol crystals, which stimulate the inflammasome, are concentrated right in the plaque. So the plaque has an engine that generates active IL-1 beta. I don't have time this afternoon to go into the myriad papers that show pro-inflammatory and pro-atherogenic effects of interleukin-1. This review gives you a key to some of this literature. The study that we are currently undertaking is using a monoclonal antibody that selectively neutralizes interleukin-1 beta, and that is currently approved by the U.S. regulatory authorities for treatment of some rare pediatric diseases that are caused by gain-of-function mutations in the inflammasome, known as the Muckle-Wells syndrome or cryopyrin-associated periodic syndrome, CAPS. This antibody has a long half-life, so that it can be given every 3 months and still retain clinical efficacy. We recently published the phase 2 data of treatment of a series of individuals with diabetes with this monoclonal antibody. After 4 months, even at some of the lower doses tested, there's a striking decrease in inflammation as gauged by blood levels of interleukin-6, a profound decrease in C-reactive protein, and very importantly, a decrease in fibrinogen levels. Remember that fibrinogen is the precursor of thrombi. So that phase 2 study sets the stage for a 17,000 plus person study, a phase 3 trial that is called the canakinumab Anti-Inflammatory Thrombosis Outcome Study, or CANTOS. CANTOS is a search trial, like I showed you in that original design, that is enrolling survivors of myocardial infarction on full panel of standard of care, including high-dose statin, but with persistent inflammation as shown by a C-reactive protein of greater than 2 milligrams per liter, and randomizing these individuals to placebo or 3 doses of this monoclonal antibody following up for hard cardiovascular events. This study currently has about 6,000 people enrolled, and I'm very proud to say, in keeping with my title about going global, is that Brazil is, as it was in the case of our Jupiter study, a very important contributor to the CANTOS trial among 40 countries that are helping us with this very important clinical experiment. So we're very excited about CANTOS. We believe that IL-1 beta antagonism is very promising in atherosclerosis because it is selective. It is an important pro-inflammatory mediator, but is not essential for host defenses. It leaves interleukin-1 signaling intact. It is implicated in atherogenesis by many preclinical studies, and it has some potential modest beneficial metabolic effects, such as a slight lowering in hemoglobin A1C. So for us, CANTOS provides an exciting opportunity to, number one, test the inflammatory hypothesis of atherosclerosis, which is a road that we started many years ago, but also, and very much in the spirit of Jorge Pinto Ribeiro, to provide a novel therapy to address residual risk in secondary preventions in our patients with atherosclerosis. If you have any interest in learning more about these trials, here are the internet addresses where you can seek further information. We're very appreciative for the support from many sources that allowed us to engage in this work and are allowing us to pursue these clinical hypotheses as well. And one of the great joys, one of the enduring joys and satisfactions of our research enterprise is the ability to collaborate with individuals all over the world. And this slide mentions a number of the key collaborators for the work that I spoke about today. But we're very proud that our trials are international and provide an illustration of how cooperation among different nations can advance the human condition and promote human health at a time when we desperately need to be reminded of that. So I would like to thank you very much for the privilege of participating in this symposium dedicated to the memory of a great man. And I would be pleased to answer questions, and I would be pleased to try to answer questions that are asked also in Portuguese. Thank you very much.
Dr. Libby, your conference fitted very, very well in our symposium, since you gave a full example of the integration between the basic science, mechanisms of disease, development of new treatments, and now going to the clinical trial. This is the main proposal of the translational research, and this is such a nice example of that. And all together with the large amounts of information that have been bring about by your team. One of them now everybody needs to know, with a colleague, graduated medicine, that's, excuse me, Marina Beltrame Moreira, that's working with you in your lab, was a student of Sandra, gets his PhD here and is doing his postdoctoral training in the laboratory of Dr. Libby.
Put up the sound, I can try to engage.
I didn't understand. I think that's complaining, está se queixando da imagem.
Is on mute, so I can't hear anything.
Não está ouvindo nada. Jorge, agora deu?
Now I can hear. Thank you.
Estás ouvindo agora, Dr. Libby? Está ouvindo agora? Estou perguntando em português porque ele fala português. Aprendeu com...
Mas eu não entendo tão bem o sotaque gaúcho.
Just repeating briefly what I said before. You don't understand the part that I... Excuse me. I have a lot of people doing signals to me here. Flavio, don't do that. Wait a minute. Let's try to talk just we. Are you listening me?
I can now hear you. Yes,
okay.
Now I can hear you.
Okay, thank you. I'm going to use that key that pilots from planes used to do, cambio, to change the words. Dr. Libby, I don't know if you listened before when I thank you very much for your very insightful conference. That was an example, a major example of the translational medicine, coming from the lab to the clinical scenario, investigating mechanisms of disease, new drugs, new treatments, and are going to be tested, are being tested right away in clinical trials, as the monoclonal antibody that's been tested in the Canto trial. We hope to have good news about this new drug in the future. It's open to questions to the conference. And Dr. Libby said that can be done in Portuguese, if someone wants to ask. Sandra.
Professor Libby, I won't challenge your ability to listen the Gaúcho accent, because it's really hard. Let me try in English. What's to be done in the future? Because you just mentioned, talked to us about your new clinical trial showing a very nice hypothesis to be tested. It enrolled or will be enrolled 17,000 patients. What it's for us to do, besides to take part as a collaborative center? What else is necessary to do right now?
Well, I think that we're going to need many opportunities to test the inflammatory hypothesis. There are probably 40 interleukins. There are probably as many chemokines. There are many different inflammatory mediators. So I think the inflammatory response is so redundant that we may need to test a number of different approaches. So we're starting here 2 trials, one with methotrexate, one with the anti-IL-1 beta antibody, but I think it may require global cooperation to test other targets in similarly designed studies, because what we're looking for is the sweet spot where we target a pathway which is causally involved in atherothrombosis without impairing host defenses against tumors and against infectious organisms. So we need to be very careful and look for opportunistic infections and impaired tumor surveillance, as sometimes happens with the anti-TNF antibodies. So I think certainly we eagerly would welcome collaboration with the CANTOS trial, which is a global trial, the methotrexate trial, which is sponsored by the U.S. National Institutes of Health, is limited to North America, not by us, but by our funder. But I would encourage people to think of their own favorite targets and to try to put together clinical trials, because it's going to take teamwork to test this hypothesis fully. I think it would be very lucky if we have a positive result with the first or second trial.
Okay, thank you. You can count on us, okay?
Please, someone have another question here in the audience? Dr. Libby, this week came out in New England, I think by the first time that a new drug had a benefit, very nice effect in patients with asthma. Again, a monoclonal antibody in the line of the inflammation reasoning that they are blocking, and the prevention, the control of asthma improved a lot with this kind of drug. In many situations, we are seeing that. This is the, I think, spectacular development in the way that we treat disease using this kind of drugs. Surely your conceptual hypothesis that drug is going to work in patients with coronary heart disease. Do you have some thoughts about the use, not as a secondary prevention, but as a primary prevention as well in the future of these monoclonal antibodies?
Right. Well, I think that if we are able to use inflammatory biomarkers to identify subjects who would be benefited by anti-inflammatory therapies, that could be a contribution with primary prevention. Anything that we target in that manner, though, would have to be beneficial on top of statins, because the Jupiter trial, which of course was conducted with the aid of our Brazilian colleagues, showed that if we took people without known cardiovascular disease with below median LDL levels but above median C-reactive protein, that if we gave them 20 milligrams daily of rosuvastatin, that we would reduce cardiovascular events in primary prevention. So we already have an intervention which is anti-inflammatory, which works in primary prevention when targeted by a biologically directed biomarker. The revolution which you pointed out is actually a twofold revolution. E não somente identificar como alvo de terapia a inflamação, mas também o uso dos fármacos de uma nova classe, os biológicos, tais como os anticorpos monoclonais. Então, eu acho que no caso de dislipidemia, os doentes que têm risco cardíaco com níveis de LDL acima do normal, apesar do tratamento com as estatinas, eu acho que os anticorpos monoclonais que tem como alvo PCSK9 vão revolucionar o tratamento destes doentes. Então, estamos na porta de uma época quando temos novos fármacos biológicos para aumentar e ajudar a aumentar os cuidados para os doentes e aumentar os fármacos que são disponíveis para a prevenção cardiológica.
Dr. Libby, as you see, his Portuguese is much better than my English. And I have studied English for now almost 7 decades, and I didn't learn yet. And Dr. Libby learned to speak Portuguese, not living in Brazil, not studying Portuguese, just talking with a guy speaking in Portuguese. That's terrific. Well, Dr. Libby, I would like to thank you very much again for your contribution to our symposium. Even that Jorge did not work on this field in terms of basic investigation, your model in the way that you see the problem and the way that you act, that you take actions in the area of research and also as a leader of teams. These characteristics have much to do with Jorge. Jorge had really that kind of profile, and there is in this room a lot of people that is in some way, at least in part, descendants, sons of Jorge. thank you very much. I hope to see you in Brazil, maybe in the next future. Your word, please.
Eu vou ficar no Congresso da Sociedade Brasileira de Cardiologia no Rio no mês de setembro.
Thank you. Bye-bye.